Hello, today I will be talking about DNA replication, how to draw the bubble, the enzymes that go in it, and the difference between eukaryotes and prokaryotes. To begin, I have written down the template of DNA with the origin in green, and helicase will start the replication, initiate it, by breaking hydrogen bonds, first starting at the origin and eventually moving out to the ends of the replication bubble at the edges of the fork. DNA gyrase will then be on the outside of the helicase. It will reduce the strain in the helix outside of it, and will occasionally make single-stranded breaks in the DNA. And the single-stranded binding proteins that I'm writing in pink are going to be on each strand to try to keep the DNA template from rejoining and binding again. Now remember that DNA is always created from the 5' to the 3' end. So if you were to write out the DNA that's synthesized in arrow form, let's start at the origin and move towards the 5' of the template. This is because DNA is, remember, anti-parallel, so the 5' end of the new DNA strand is actually starting at the origin for the bottom template and the origin again at the top template. And DNA is going to be moving in both directions because you have two ends of the replication fork. And so if we keep that going in both directions, you're going to have Okazaki fragments that I'm writing here. Okazaki fragments exist because you're going the opposite direction in which DNA is wanted to be made. And these fragments are helped by the initiative of primase. So primase will work at the beginning of the origin, where the leading strand is, and will work from the helicase site for the Okazaki fragments to form. Remember, primase creates RNA, not DNA. The RNA primer is needed for DNA to be created. DNA polymerase III always needs the RNA to start. So if primase doesn't work, DNA pol 3 won't elongate the DNA and you will never have new DNA. So what I'm writing in blue is DNA created by the polymerase III. And after the polymerase III is elongated, pol 1 will replace the DNA. The DNA pol 1 will replace the RNA with DNA. And then finally, once that has been done, the DNA ligase can come in and attach, like glue, all of the fragments, including the leading strand, to make one piece of DNA. And then I'm going to write it in red. Right. So next, after this, I'm going to talk a little bit about the differences between prokaryotes and eukaryotes. DNA polymerase I has no equivalent in eukaryotes. Instead, eukaryotes use a bunch of different enzymes to achieve the same thing as DNA pol 1. DNA pol 3 is replaced by DNA polymerase delta in eukaryotes. And primase in prokaryotes is replaced by DNA pol alpha in eukaryotes. And 3' to 5' exonuclease activity, that's in the opposite direction of DNA synthesis. Synthesis is from 5' to 3'. In prokaryotes, pol 1 and pol 3 work in the backwards direction to make corrections to the DNA, and in eukaryotes, delta does this. Now, in the forward direction, in prokaryotes, pol 1 works, and in eukaryotes, it's the other enzymes that do the exonuclease activity. Remember, pol 1 replaces the RNA primers with DNA, and so do these other enzymes in eukaryotes. In prokaryotes, there's one origin. This is where the initiation always starts. In eukaryotes, there's many origins, and this helps the eukaryotes replicate DNA much faster than they would if they only had one. And each origin has licensing factors attached to them, and those licensing factors control how many times the DNA gets replicated to ensure that it only occurs once. Termination occurs in prokaryotes when the DNA strand, the new synthesized one, meets the origin once again. In eukaryotes, it happens when the synthesized DNA, meets the telomeres, which are the ends of the chromosomes. And remember, telomeres are short tandem repeats, and they're also non-functional, meaning they don't code for any genes or something that is functional. In eukaryotes, termination also occurs at the end of the Okazaki fragments. This is because the Okazaki fragment meets the other Okazaki fragments, and there's no more template for the DNA to be replicated. Alright, I hope this video helped a little bit in understanding how to draw the replication bubble, and a little bit more about the differences between eukaryotes and prokaryotes. Thanks.